classification. The father of classification, or father of taxonomy, is Carolus Linnaeus. He was the guy that came up with the system that we use today. There are many other scientists out there using some sort of taxonomy. Linnaeus had the one that was simplest and easiest to use by most people. It's called binomial nomenclature. With binomial nomenclature, you have two names. You use the genus and the species name. When you're typing out the genus species name, you're going to use italics. When you are printing it out yourself or writing it yourself, you're going to underline. The genus is going to have a capital letter. The species is going to have a lowercase letter. He also started a hierarchical grouping of organisms where he put organisms into general categories. Back in his time, for the most part, was kind of a plant animal sort of classification. Over time, it's changed several times, going from 2 kingdoms to 6, 7, 8 kingdoms to 3 kingdoms, and kind of back and forth. Today, we have domain being the most broad, and there are 3 domains. This contains the most organisms, all the way down to species, the one that is most exclusive. We start with domain, then to kingdom, phylum, class, order, family, genus, species. You might remember the saying, King Philip came over from Germany soaked. There are several others out there. That just tends to be the most common. Species is the most exclusive. It's the one that has the least amount of organisms in there. So the species is the one that can only interbreed with itself. Here is our domain system. We have bacteria. Bacteria is a single-celled organism. It's a prokaryote. Then we have archaea, and if you notice, archaea, it is a single-celled organism. It is not necessarily classified as a prokaryote, and it is not a eukaryote. It's an in-between. And if you notice on the tree of life here, that archaea are more closely related to eukarya than it is to bacteria. So bacteria and archaea and eukarya have a common ancestor of down here, and archaea and eukarya have a common ancestor much closer to each other. Archaea are organisms that live in extreme environments. These are ones representative of the most primitive organisms that were on the planet. It is a little bit surprising that they are more closely related to eukaryotes than bacteria, and scientists have figured this out through the use of mitochondrial DNA testing. Then the last one is eukarya or eukaryotes. There are 4 kingdoms under the eukarya domain. We have protista. These are a kind of laundry list of organisms, the ones that are misfits that don't fit anywhere else. They range from single-celled organisms to large multicellular sea kelp organisms. We have plants, fungi, and animals. Phylogenetics is the study of evolutionary relationships among organisms and then grouping them according so. This is the old tree of life. This was from 1866, grouping things together in a tree-like formation. And this is what we have come up with today of all the different organisms branching on our tree of life. In the red and the green are the eukarya and the archaea, and the purple is the bacteria. Cladistics is a way to group organisms according to characteristics. Generally, we look to the common ancestor to find these derived characteristics. Looking at this picture here, you can see over time the change of that organism in order to make our new species. Here is the common ancestor, and as generation after generation there were variations and changes, you can see how new species have arose and how the common ancestor relates to those new species. This is the most generalized tree for the eukaryote tree. How we actually make a cladogram. is first we need to find all the characteristics that are in common. You're going to group together the species with the most characteristics in common. There's something called parsimony that you need to follow when you are looking or when you're making these groups, and that's that the simplest explanation is going to be true. Then there's also the maximum likelihood that the more closely related the organisms are, the fewer genetic sequences. There are some assumptions that go along with these two ideas, but when you're making a cladogram, you want to have the least amount of explanation to get there, and then the ones that are more closely related have fewer differences in their genes. This is just 5 different ways that you can make a cladogram look. When you are forming your cladogram, each time there's a branch, there's a node. So this is a node here. Then you have the tips. This is where the species lies. And the branches are the lines, are the lineage of that organism. Here we have 2 cladograms. We have one with the mammals and the birds more closely related, and the second one with the birds and the lizards more closely related. If you take a look at the bottom one, we have 4-chambered heart and 4-chambered heart. So here where we have too many differences on the chart, where up here we just have the one change. So this top one is exhibiting parsimony because it is the simplest explanation for what is happening. The bottom one does not have parsimony because there are too many differences or too many steps in order to get to the end product. You can make different groupings with your cladogram. A monophyletic grouping is when all of the descendants in that grouping are from one common ancestor. Paraphyletic here, where the grouping is from, or some of the descendants are from one common ancestor, some are not. And then polyphyletic here, when we are from more than one common ancestor. Because we have here and here as our more than one common ancestor. And here's what you should know.